breeding crops for biotic and abiotic stresses. Biotic stress is damage caused by living organisms, pests, pathogens, weeds, etc. Abiotic stress is damage caused by non-living organisms, drought, flooding, etc. These stresses produce significant impact on crop yield and quality. Importance of breeding for stresses. The loss in global food production due to environmental stresses threatens food security and livelihoods worldwide. To combat this, developing resilient crops is essential for sustaining agricultural yields and adapting to changing climate conditions. Traditional methods like pesticides and chemical controls are costly and lead to environmental degradation. Sustainable alternatives are increasingly necessary to protect ecosystems and ensure long-term agriculture. Sources of resistance. Wild relatives of crops serve as vital source of genetic variation, offering resistance genes that can enhance crop resistance to pests and diseases. Harnessing this natural diversity is essential for developing robust, sustainable agricultural practices. Modern genetic techniques like molecular breeding, CRISPR-Cas9, and transgenic approaches enable precise modifications in crops, enhancing traits such as yield and resistance. These innovations offer promising solutions for addressing global food security challenges in a rapidly changing environment. Classical breeding approaches play a crucial role in enhancing crop resilience through several key techniques. Crossbreeding involves introducing resistance traits from naturally resistant varieties into susceptible ones, allowing for improved disease and pest resistance. Back-crossbreeding further refines this process by ensuring that desirable agronomic traits, such as yield and quality, are preserved while integrating resistance characteristics. Additionally, marker-assisted selection involves molecular markers linked to specific resistance genes, streamlining the breeding process and increasing the precision of trait selection. Together, these methods form a robust framework for developing resilient crops that can better withstand environmental challenges. The development of crop varieties resistant to specific pests and diseases is essential for improving agricultural resilience and productivity. For instance, efforts to create wheat varieties resistant to stem rust, particularly the UG99 strain, are critical for protecting wheat production worldwide. Similarly, Bt cotton has been genetically modified to effectively resist cotton bollworm, reducing reliance on chemical pesticides and improving yields. In rice cultivation, breeders are focusing on developing varieties with resistance to fungal diseases like rice blast, which can devastate crops. Additionally, the creation of drought-resistant maize varieties in sub-Saharan Africa addresses the pressing challenges posed by climate change, ensuring food security in regions prone to water scarcity. Collectively, these advancements illustrate the importance of biotechnology and traditional breeding methods in cultivating resilient crops to meet the demands of a growing population. Modern genomics approaches are revolutionizing the way we enhance crop resistance through precise and innovative techniques. CRISPR-Cas9 enables targeted genome editing, allowing for the efficient insertion of resistance genes into crops, thereby enhancing their ability to withstand pests and diseases. Meanwhile, genomic selection employs genome-wide markers to predict and select for resistant lines, streamlining the breeding process and improving the accuracy of trait development. Additionally, transcriptomics and proteomics provide valuable insights into stress-responsive genes and proteins, helping researchers understand how plants respond to environmental challenges at a molecular level. Together, these advanced methodologies offer powerful tools for developing resilient crops that can adapt to a rapidly changing climate and shifting agricultural demands. Breeding for durable resistance focuses on enhancing crop resilience by integrating multiple resistance genes, a strategy known as pyramiding. Gene stacking further enhances this strategy by using biotechnology to insert several resistance genes simultaneously, ensuring that crops can withstand various threats. The field of plant breeding faces several challenges, notably the evolution of new pathogen races that can overcome existing resistance, which threatens crop productivity and food security. Additionally, there is often a trade-off between resistance and yield, where enhancing one trait may inadvertently compromise the other, complicating breeding strategies. The complex inheritance of resistance traits further complicates efforts to develop durable varieties, as multiple genes and environmental factors can influence outcomes. Looking ahead, future directions in breeding research should focus on integrating advanced phenotyping, machine learning, and omics technologies to enable precision breeding. These innovations promise to enhance our understanding of plant responses to stress, streamline the identification of desirable traits, and ultimately lead to the development of resilient crops capable of thriving in changing environmental conditions. Stress resistance is vital for achieving sustainable agriculture. The integration of traditional and modern breeding approaches, alongside ongoing research investment, is essential for developing resilient crops and securing global food security.